almost everybody, so I'm going to get going. So we're going to pick up this morning with where we left off on last Thursday, walking through a couple of real-world analyses that we can do with BEDtools. And today we're going to focus specifically on this question of how we know, this is a common problem in genomics, how we know if an observed number of intersections or overlaps, like I have some transcription factor binding peaks, and I have some other genomic annotation. I observe overlaps between them, suggesting some sort of biological relationship. But how do we decide whether or not that number of intersections or that relationship is statistically significant? And another way of thinking about whether something is statistically significant is to ask, is the observation more extreme than we would expect by chance? And it's that expectation by chance that becomes a bit tricky sometimes. So that's what we're going to focus on today. But before I jump in, I just want to make an announcement that Sarah and Gage are going to switch office hours this week. I forget who's on Wednesday and who's on Friday. Sarah or Gage, could you just jump in and explain the switch? Just so everyone...
Usually I'm on Friday, Sarah's on Wednesday. We both have things during our time, so we are going to switch this week. So just use the same link though for the regular TA hours for that day, and we'll just join that one.
Great, thank you. Okay, so again, we're going to be doing a little bit of work on Malibu today. So if you could VPN in and then SSH into Malibu. We are going to be playing around with the same files that we worked on last time. So these are these enhancers from human embryonic stem cells, and we're going to bring in some new data sets today. So to get at this common question of testing whether or not an observed number of intersections between two data sets, say transcription factor and binding sites and enhancers, is significant, we're gonna focus on the following question. Last time we identified enhancers in these human embryonic stem cells. What we're going to ask today is whether or not single nucleotide polymorphisms, or Snips, identified from genome-wide association studies, or GWAS, overlap enhancers in human embryonic stem cells more often than expected by chance. And just to set this experiment up a little bit, if you're not familiar with genome-wide association studies, the basic study design is the following. You collect a whole bunch of individuals, patients with a particular phenotype, and then you have, those are cases, and then you have a ideally equally sized collection of control individuals without a disease phenotype. You study the genetic variation in those two groups, and you're looking for single nucleotide polymorphisms or genetic variants that are enriched or are more common in the case individuals and in the controls. And the inference is that those genetic variants comprise a genotype that is associated with the disease phenotype. And that type of study design is still popular today, but it's especially popular sort of 1998 through 2012, 2013. And it was typically done with a technology called SNP arrays, whereas instead of sequencing an entire genome, you would actually had an array that had oligonucleotide probes that would interrogate, say, 100,000 or 500,000 known genetic variants in the human genome. By and large, what those genome-wide association studies found is genetic variants that are largely not in protein coding genes. And so, and those genetic variants that are sort of upstream of genes or intronic within genes that are associated with these disease phenotypes. The work that's been done over the last, say, 10 years is to try and understand, well, if they are not in protein coding genes, what is it that associates these genetic variants with disease phenotypes? Is it the fact that these genetic variants regulate the expression of downstream genes, or maybe they're just markers that are genetically linked to other genetic variants that are within the genes that just aren't assayed by these SNP arrays? That's neither here nor there for this question, but the reason we're asking this question actually is there is a study from the ENCODE project that's characterized transcription factor binding gene expression and tried to understand sort of the regulatory network of gene expression in various tissues. One of their key findings is that they show that actually the SNPs identified as associated with disease phenotypes through GWAS studies tend to overlap enhancers more than expected by chance, suggesting that the reason that these genetic variants are associated with disease is because of some impact on gene regulation during development or later on in life, or in particular tissues that are relevant to the disease. So this question is really going after trying to reproduce that result. Let's see if we can actually see the same signal. All right. So to do this, again, we need some materials to do this. We need human embryonic stem cell enhancers. We've got those on Malibu already. We downloaded those. We need a catalog of significant single nucleotide polymorphisms from genome-wide association studies. It turns out that there's a catalog maintained by the NIH of single nucleotide polymorphisms and the disease traits to which they are significantly associated. So essentially, we're going to download a file that represents that. Now, what we need to do is we need to measure the number of observed overlaps between the two. So how many of these genome-wide associated SNPs, or these SNPs from GWAS studies, overlap enhancers? Now, the question is whether or not that observation is more or less than what we would expect by chance. And when I say what we expect or expect by chance, what we really mean is this observation sort of close to the mean or within the expected range of a distribution, or is it way out in the tails of a distribution? And if it's way out in the tails of the distribution, that suggests that maybe the observation is actually interesting, whereas if it's sort of in the center of a distribution, say the normal distribution, if it's less than one standard deviation from the mean, then maybe it's not so interesting. Maybe this is exactly the type of overlap we would expect to see. Now, the challenge with defining an expectation with overlaps is it's not clear what the underlying probability distribution would be. It's not necessarily, we don't know that it's Poisson, we don't know that it follows a normal distribution. So what is typically done is a Monte Carlo experiment where you essentially iteratively shuffle these genomic features, or randomly put them in random locations in the genome. And for each time that you randomly shuffle those intervals, you then measure the intersection, and you do that over and over again. Each time you shuffle, you measure the intersection and you see how many overlaps there are. And that can help you define a null expectation, where the null is just like, okay, if these SNPs and enhancers were just in any old place in the genome, what would it look like? And that can help you to compare what you observe to what you might expect. And so we're going to talk through how we go about doing that and some caveats associated with this. Okay, so first of all, we need enhancers. And this time I just decided to make it a little more interesting by including not just the strong enhancers, which we used last time, but the strong and weak enhancers. And this is mainly so that we have more enhancers throughout the genome to compare to GWAS SNPs, because I think with the strong enhancers in human embryonic stem cells, it was only like 400 or 500 that were reported. Okay, and so we can just grep for now just enhancer, and that will pull out weak and strong enhancers from this bed file. This little sed trick is to remove the chromosome label at the beginning of the chromosome. So we have 1 and 2 and 3 instead of CHR1, CHR2, basically so the bed files match. All right. So I'm going to take this command and run it. Oops. Let me just make sure I have the file I need here. I don't, because I made some new directory. What was it called? BEDtools demo. All right, so I've made this file called human embryonic stem cells, chromHMM, all enhancers, no chromosome labels.bed. Let's just take a quick peek at that. Right, and this looks not surprisingly very similar to what's on the screen. All right, so now we have a bed file of all enhancers in human embryonic stem cells. Now what we need to get is a catalog of significant Snips from the UCSC genome browser. I have this file already stored up on AWS. I just want to show you quickly as a reminder how to do this from the UCSC genome browser. Tools, table browser, build 38, group would be phenotypes and literature. And in here, there's GWAS catalog. And then within there, I think it's this. So let me just take a look at the table. Right. So here's some sample data. You have a chromosome, start coordinate, end coordinate. All of these are length 1 because it's a single nucleotide polymorphism. Most of these have a so-called RSID. This is a reference to a database that the NIH maintains called DB-SNP, the database of single nucleotide polymorphisms. And for each SNP, it's usually tracking a PubMed ID and the author, and then gives you a sense of what phenotype, well, the title of the paper rather, and then what phenotype the SNP is associated with. Okay, so that's how you can go get this for yourself. I actually stripped off a lot of that, almost all that information, just to keep things simple. So if we copy and paste that wget command to download the file that I created, I basically took the file from UCSC and I just kept the RSID, the PubMed ID, and then that trait. So there's all sorts of traits in here. Here's a SNP that's associated with people who are morning versus evening people, I think. I guess that's what a chronotype is. Here's one that's associated with BMI, colitis, IBD, lots of stuff, okay? We are gonna take all SNPs. We don't really care about the phenotype. The question that we're going after is really, are genetic variants identified from GWAS studies enriched globally in enhancers? Okay. All right, so it's a bed file. Oop, we have to actually do this again. We have to strip off the CHR. As you can see, the CHR is at the beginning there. So let me steal this command. That's funny. It also kind of butchered my header a little bit. I removed the CHR from my header. Well, we'll live with that. A little bug. All right, so we've got our single nucleotide polymorphisms and we've got our enhancers. Now we need to intersect them. We want to figure out what our observed value is. And again, we have to compare to an expectation. Essentially, p-values are nothing more than a reflection of whether what you observe, the quantity that you observe for a given random variable is the probability that you would see that by chance. So we're going to develop our observation now. To derive a p-value, we have to figure out what the probability of observing what we observed by chance. And that's where the Monte Carlo simulation will come in. Okay, so let's just figure out how many, first of all, let's just see how many data points we're looking at. The command's right here. I'm not going to do this on Unix. But if we look at the number of GWAS SNPs, there's 39,000 of those. And now when we've included weak and strong enhancers, we've gone from 400 enhancers up to 240,000. Okay, so two fairly large collections of genome intervals. And what we're going to ask is how many of the SNPs overlap enhancers. And the reason we're using this -u option at the end, this is -u means unique. That means if a GWAS SNP overlaps one or more enhancers, I just want to report at once that yes, there was an overlap, so that we're not double counting. Because if there's one GWAS SNP that overlaps 1,000 enhancers, we don't want that to inflate our statistic. If the question is what fraction of GWAS SNPs do overlap an enhancer. Okay, so let's run this command, double check that we get the same result. Okay, we do. Let me just show you. Let's just take a look at what these look like. So yeah, here's a SNP that overlaps an enhancer, and you can see that this enhancer is 400 nucleotides long, and yeah, this SNP is somewhere in the middle there. Okay, but here's an example of a SNP that overlaps multiple enhancers. Yeah, there's actually the same enhancers reported multiple times in the BED file for enhancers. So we want to just collapse this down to one hit to say that yes, this SNP does overlap an enhancer. Okay, and that's what the -u is doing. Just take a look at that. So this just gives us a unique list of SNPs that do overlap enhancers. All right, so that's our observed. So 2,040 out of the 39,335 SNPs overlap an enhancer. So that's 5.2%. So the question now is that interesting? Is that more than we would expect by chance? So how do we know? That's really where we're going now. A common strategy for figuring out how we know is to do what's called a Monte Carlo simulation. So we really want to compare what we observed, which is 2,040 intersections, to what we expect, to assess whether or not that 2,040 or 5% of SNPs is significant. And when I'm talking about an expectation, we're really talking about a null hypothesis. Like, what is the null distribution of overlaps between GWAS SNPs and enhancers? And there's a couple of common ways. There's two common ways to get at this. If we know that the intersections follow a normal distribution or follow some distribution that we can just plug into a formula using R, then, and we know the parameters of that distribution, such as the lambda for the Poisson distribution or the mean and standard deviation from a normal distribution, we can directly just plug and chug our numbers directly into R and get a p-value. So that's one way. It's common, however, for this type of analysis to derive what's called an empirical p-value. And that's the empirical, meaning that we actually are building up our expectation manually, because the distribution of intersections in this case is not, we don't really know what the underlying distribution is. We basically just build up the distribution directly ourselves and compare what we observe to this null distribution or null hypothesis. And that's where this Monte Carlo simulation comes from. And why is it called Monte Carlo? And that gets at the history of Monte Carlo being a location where people go to gamble, and this notion of chance, probability, random chance. So the way the experimental design to do a Monte Carlo simulation for genomic overlaps is, as I mentioned before, to repeatedly, namely thousands of times, shuffle the location of every one of the GWAS SNPs into random locations of the genome, part A. Part B, shuffle the intervals from all the enhancers randomly throughout the genome. They've been each have been shuffled. Now we run the intersection again on the shuffle data set and ask how many overlaps there are. And so for each of the thousands of simulations, we get an overlap statistic that reflects the probability, that reflects the number of overlaps when the intervals are randomly shuffled. So if we do that thousands of times, essentially what we're going to build up is a histogram of the number of overlaps observed when shuffled. And and the reason we do it thousands of times is so that that histogram is well formed. It's nice and smooth, and we've done lots and lots of trials to really measure the extremes of what that null distribution could look like. If we only did this experiment a couple of times, obviously we're gonna have a really ragged, not very informative histogram against which to compare our original observation of 2040. Right, so just to show you what this looks like, I might have to run this a couple times. A guy named Ryan Layer, who was a star postdoc in my lab a couple years ago, made this animation to show what's actually happening with a Monte Carlo simulation. So let me see if I can get this to work. Okay, so what you're going to see are two sets of hypothetical intervals. In yellow, you're going to see the GWAS SNPs, and in blue, you're going to see the enhancers. And what's going to happen is these intervals are going to be shuffled. You're going to see them moving all around. And on the right, there's going to be a histogram, which will show the number of observed intersections that were seen after you did the shuffle for each of these Monte Carlo iterations. So let's just see if we can get this to run. Okay, so in this case, he's actually just shuffling the GWAS SNPs. The location of the enhancers remains the same. And those vertical white lines show where the intersections are when they're shuffled. So let me play it again. And for each iteration, give this one more try. Okay, you can see on the histogram, the x-axis is the number of intersections observed, and the y-axis is the number of Monte Carlo trials that had that number of intersections. So this distribution is our null. This is our null hypothesis. So the expected value from this Monte Carlo simulation is something like, I don't know, 4.5. That looks like roughly the mean, or maybe even 5, because most of the Monte Carlo simulations, when shuffled, there was 5 intersections. Rarely were there less than 1 or less than 2. And the most number of intersections that was observed, this is just a toy. We'll have to do this on our own. But in this simulation that Ryan did, the largest observed intersections when shuffling was 9. So essentially what you could do is then, all right, let's say in this hypothetical case, you observed 100 intersections. Well, 100 is way out to the tail. It's much higher than you'd expect by chance. And so from that, you could actually derive a p-value. Okay, and I'll talk about actually what that p-value is in a second. Any questions about this so far? This is kind of a fundamental concept in really, well, in genomics, but in lots of different scientific disciplines where when you don't know what the underlying distribution is, you derive this empirical p-value of Monte Carlo simulation. Does it make sense so far? Okay. Well, how do we go about doing this? Well, we're going to learn how to do a for loop in Bash. We're going to actually compute this Monte Carlo simulation now. Okay, so I've cheated and helped myself with this for loop because I always get them wrong. Save myself a little Google here. But what we're going to do, let's just walk through what this code is doing. We're doing a loop that will go through 100 times. There's a Unix command called seek, which will generate a set of numbers from 1 to 100. So I'm just show you this. Okay, seek 1 to 1,000, seek 1 to 10, 1 to 2. Okay, so this for loop, it runs this command between the backticks, which generates a sequence of numbers from 1 to 100. So the way the for loop in Bash works is if you feed it an input of 1 to 100 or lines in a file, it's going to iterate. It's going to do the operations that are within the for loop that many times. So if I change this to 20, it's going to execute these commands in the middle here 20 times. So essentially this command that's between the backticks gets fed into this for loop. And so this value I takes the values 1, 2, 3, 4, 5, all the way to 100. And for each value of I that's dictated by the sequence that we're generating, you say do, and whatever's between this do and done block gets executed I times, or whatever the number of iterations in the for loop are. Okay, so what gets executed? Well, the first step is we're going to shuffle the GWAS SNPs to random locations in the genome. And in this case, we're doing a simpler version of the Monte Carlo where we're just shuffling one set of intervals and we're keeping the other set of intervals, the enhancers, in place. So we're just randomizing the location of the GWAS SNPs and comparing to the enhancers. We take the shuffled locations of the GWAS SNPs and we pipe those into an intersect command. And in this case, we say that the A file is coming from a pipe. That's what this dash means. And we're intersecting those shuffled GWAS SNPs again with the enhancers. And we pipe that to WC-L, which will count the number of intersections. Okay, so let's just see what that looks like. Looks like I need a genome file. It looks like I renamed something different. What did I call it last time? I feel like we had a genome file before. Okay. Well, I'll just change it here. It's genome.txt, at least for me. You might have a file called human.grch37.no-chrome from last time. I somehow deleted it if I actually created it last time. So it's either this file or if your directory looks like mine, it's just going to be genome.txt. Okay. Change that. Okay. So this doesn't like the fact that, So we have a problem, and it's warning that we have a problem because the genome file uses CHR for the chromosomes, whereas our data files don't. So I did something last time to create this genome file properly. So I need to go back in history and look at what I did. So I need to strip off the chromosome label again. What did I call it here? Should be in business now. Okay, so the reason I need, first of all, the reason I needed this genome file again is if you're asking BEDtools to shuffle these intervals to random locations of the genome, it needs to know the length of each chromosome so that it knows, you know, how many options it has in terms of placing intervals in one chromosome versus the other. So essentially what it's doing concretely is for each interval in this GWAS file, it's looking at the length, all the length of the genome or length of each chromosome and choosing a random number for each, it chooses a chromosome randomly. And then for that chromosome, it chooses a random number between 0 and the length of that chromosome at which to put randomly shuffle the interval. Okay, so we've shuffled all the GWAS SNPs. And then for each of those shuffled SNPs, we're intersecting it with the original enhancers. So let's see, for one, this would be one Monte Carlo trial. We're shuffling and counting the intersections. So that in that trial, we saw 1,346 intersections. It's random. The next trial is going to be different because the shuffling is choosing random locations. The next trial, 1,403. The next trial, 1,379, 1,364, and so on and so forth. So if I wanted to do this simulation, I could run this command on the command line as I'm doing 100 times, right? And each time I could copy this number and I could paste it into a spreadsheet and, you know, spend an hour of my life doing that. I don't recommend that. It's not terribly reproducible. And that's where this for loop really helps us. So this for loop is taking the same thing, but now what we're doing, it's running this -wc-l command to get that number. But here's the special trick at the end. That number is appended. This greater than sign means not only create this file if it doesn't exist, but for each iteration, append. That's what the extra greater than sign means. Append the current number to this file. So you build up a list of all the intersections. Okay, so let's run this for loop now. And this will take a second, more than a second. It takes about probably a second or two for each iteration. And you can see that when this is running, it takes a while. My command line was held up. I just got it back because it finished. I want to show you one trick that I don't think I've showed before. If I just put an ampersand at the end of this command, it will run this experiment in the background and give me my command line. So let me just delete this file so we can see this anew. So if I run this command and put an ampersand at the end, what you see is I get my command line back. And if I go to the bottom, if I do ls-ltr, you can see that this file is growing. First it was 165 bytes, then 210, then 235, 485, and now it's finally finished. So that ampersand, when the job is done, I submitted that program or that loop as a job or a process. When it's done, it'll pop back up on the screen and say, ah, that's done. And then we know that we can go take a look at the output of this file. So how many lines should be in this file? Anyone have a guess?
100.
100, right? Because we did 100 Monte Carlo simulations. All right, there are 100. That checks out. I said we should do thousands typically, and you should. I just did 100 because I would like this to finish the time that we have in the class. So here's all the number of intersections that were seen when we shuffled the genomes for each of the experiments. Okay. This is giving away a problem from last time. I set it up before where there was a chromosome labeling issue. So all the experiments gave 0 overlaps. I've fixed that ahead of time now, where we actually get non-zero overlaps for each experiment. Okay. So what I want to talk through now is how we consider whether or not our original observation of, let's see, 2040 intersections is significant. Is this 2040 more than we expect by chance? Well, one thing we could do is just look at this distribution. So let me see, I'm going to sort these numbers. And the lowest value is 1283, and the highest value is 1493. So the number of observed was 2040. So a common practice to derive an empirical p-value would be, in this case, we did 100 experiments. So empirically, we know that the p-value is less than 0.01, in the sense that we did 100 experiments and we never saw, with that shuffling experiment, we never saw an intersection value that was greater than what we observed with our own data, with our original observation. So that allows you to assign a p-value that is less than 0.01, because there was no simulation that had, out of 100, hence the 0.01, that had a number of intersections that was greater than what we observed. So that's a so-called one-tailed test. We're assigning this p-value based upon one tail of the distribution. We're asking, how likely is it that we would see something that is greater than or equal to our observed number of intersections by chance? Now, if we wanted to be more confident in our observation, we might want to do more Monte Carlo simulations. So if we did 1,000 simulations and we still never saw anything that was greater or equal to what we observed, then we could say the empirical p-value is less than 0.001. If we did it 10,000 times and we still saw the same thing, we'd say less than 0.0001. And if we saw 1 or 2 that were greater than what we observed by chance, then you basically, that empirical value is, say, if we saw 2 times it was greater than or equal to what we observed, then it's 2 divided by the number of simulations that we did. Okay. There's some problems with this experiment, though. Well, there's several. the biggest problem with this type of experiment, there's nothing wrong with it conceptually. Shuffling things and testing empirically how often we see, when the data is randomized, we see intersections that are similar or equal to what we observe is a totally rational thing to do, and it's defensible statistically. The problem, though, is Monte Carlo simulations assume that all of the data that you're shuffling is independent of all the other data. So concretely, it assumes that all the Snips and all the enhancers are completely independent of one another. So we know that that's not the case in genomics. We know that in many cases, for instance, enhancers tend to be spatially correlated with transcription start sites. And enhancers tend to be clustered with one another upstream of a gene. So that's already a violation of this assumption of independence. The other problem with the experiment is, let's just take a look at the genome file. The other problem with the experiment is we are giving the BEDtools shuffle command the opportunity to shuffle these genomic intervals to any one of these chromosome labels in this file. So, I think it makes sense to shuffle the chromosome 1. It makes sense to shuffle the chromosome 14. But what I'm getting at is, does it really make sense to allow BEDtools to shuffle to these so-called random chromosomes or random contigs? So what are those? These are parts of the human genome that have been assembled, and there's a nucleotide sequence for them, and hence there's a length, but they haven't been confidently placed on a particular location of chromosome 17. So this means that there is an assembled sequence that is 37,000 nucleotides long that belongs somewhere on chromosome 17, but it's not quite clear where. The reason that is probably not fair play is that those random chromosomes probably were not part of the original GWAS studies. So if these sequences weren't in the domain of the original GWAS experiment, then it's not fair play to allow them to be part of the domain of this Monte Carlo simulation. So essentially, if you were restricted in what parts of the genome you could look at in the original study, you have to restrict yourself in the same way when you do this Monte Carlo simulation. Similarly, we know for chromosome 1 that a lot of that sequence, that 249 million nucleotide sequence, is actually ends, telomeres, gaps, centromeres. It's not fair play to shuffle GWAS SNPs into the centromeres, because that's, there's no way that enhancers could actually be there. So you're going to have a depleted number of overlaps, simply because sometimes we're shuffling SNPs into centromeres or telomeres, and there are never going to be enhancers predicted there because the nucleotide sequences end.
A question.
Yeah.
Why not just resample the sites from the SNP array?
Resample the sites from the SNP array?
Yeah, because that's our effectively all the sites that have the possibility to be detected as significant.
Yes, so that you're getting into what I'm about to talk about, which is when you're doing Monte Carlo simulations, you can constrain the domain of the shuffling. And so what John is suggesting is in line with this, which is you can define parts of the genome that are in play, namely the set of SNPs that were tested. That's one thing you could do. If we want to shuffle enhancers, it's not so easy. We have to define parts of the genome that are out of play. And so one way you could define parts of the genome that are out of play would be to exclude centromeres, exclude gaps, exclude, you'd have to think about what you want to exclude, but those are some sort of easy ones to define off the bat. So Brent Pedersen was a staff scientist in my lab, and we met maybe 2012 or 2013. I was working on this problem at roughly the same time that he was, and he's got this really nice paper with Subo Day and Katerina Kekris from 2013 that really touches upon this problem. I actually would encourage you to all to read this paper if you're at all interested in this topic or think you will be doing this type of thing in your research. This is a really well done paper talking about sort of this dilemma of choosing the ideal permutation strategy. So this Monte Carlo shuffling is permutations. You're just permuting the location of all these different intervals, and how choosing that ideal permutation strategy influences the measurement of statistical significance. So what I want to spell out a bit more clearly before I get into the last couple points on this is thinking through the impact of this problem of allowing genomic intervals to be shuffled in anywhere. If we're allowing these GWAS SNPs to be shuffled onto these random chromosomes into gaps and centromeres and stuff like that, that automatically reduces the number of intersections that you're going to observe. And if you're doing a one-tailed test where you're looking for asking whether or not what you observe is greater than what you'd expect, is it way out in the tails of the distribution? Well, if what you see from the Monte Carlo simulation is if the number of intersections observed from the Monte Carlo simulations is lower artificially because of this shuffling into random parts of the genome, that is going to artificially boost the significance of the p-values that you get because your tails are not as broad as they would be. You've sort of cooked the books a little bit. So that's the mindset that this paper is about. Like, how do we handle this problem? So there's this nice figure 1 from their paper, talks about different ways that you can shuffle. So I talked about shuffling across the whole genome. You could make things a little bit simpler, but by maybe like just shuffling intervals to random locations within the chromosome that they're already on in the original data set. But what I want to talk about now is this third strategy, which is shuffling with additional constraints. So these gray regions are so-called exclude regions. You're going to say, all right, I'm going to shuffle these blue intervals, and I'm going to shuffle the orange intervals to random locations in the genome, but I'm not going to allow them to go into these gray regions, namely things like gaps or centromeres or telomeres. And that should essentially move the intersections that you observe from the Monte Carlo simulations closer to reality. It's going to make them a bit higher because you're sort of limiting the search space in the genome that things can go to. Okay. I mean, in an extreme example, imagine we exclude everything but a 1 kb window in the entire genome, and things can only be shuffled there. Well, you're going to see intersections, right? Because we've constrained the domain of where things can go. And since we've got 30,000 SNPs that got to be placed in that 10 kb window, and you've got all these enhancers, yeah, you're probably going to see intersections. But to John's point, so the BEDtools shuffle tool has this notion of -incl include regions and -excl exclude regions. So the XCL, EXCL is what I was talking about before. Like, imagine this is a big gap in the genome. We're not going to allow intervals to be shuffled there. But what John mentioned is, if you know something about the experimental design of one of the data sets, for instance, these GWAS SNPs, one thing we could do is we could go out and figure out all the different SNP arrays that were used to generate that catalog of GWAS SNPs. And let's say there's 2 million individual single nucleotide positions that represent the union of all those experimental designs. We could, if we could figure that out, and you can, that exists, you could actually say, all right, well, when I shuffle the GWAS SNPs, I'm going to define this INCL include file, which has 2 million positions in it. And BEDtools will just shuffle those SNPs within those regions. And then essentially when for each iteration of the Monte Carlo simulation, you've got an estimate or a null model that is much more closely aligned with the actual domain or the boundaries of what you could observe given the experimental design from GWAS. So that's, I think, a bit beyond what we should attempt to do in this class. But I think if you're ever doing this type of analysis, the main take home here is that you should think hard about how you define the boundaries of where you can shuffle things within the genome so that you get a much more well-calibrated empirical p-value by comparing your observed value to the empirical distribution